Hello again and welcome to part 6 of this 8-part series looking at colour genetics in budgerigars. This series supports a PDF book that I have produced. The book is available on the Shed Talk Facebook page. It's free to download, so go across, join the Shed Book Facebook page and you can download it. The PDF book contains more information, a bit more in-depth explanation of some of the key themes that we cover in this series. Well, as always, if you do enjoy the series, don't forget to hit the like button and of course do subscribe to the channel. Well, in the last two parts of this series, so part 5A and part 5B, we looked at the impact of where the gene was situated on the chromosome, and indeed we'll come back to that in part 7 of the guide, but as well as the outcome of any prediction being linked to the chromosome. So some of our mutations are impacted by the gene's locus, or the mutation's locus, and this occurs when a gene might mutate more than once. So there are more than one mutation in the same locus, and that's what we're going to explore in part 6. When genes are linked in this way, we sometimes refer to them as gene families, and in this particular video we're going to look at one particular gene family and see how this multiple allele impacts on our prediction, on how we predict the outcome of any two pairings or any particular pairing. So what we'll do now then is we'll nip over to the computer and we'll put up the presentation for this particular video, and we'll talk through multiple allele. So before we start to look at the multiple allele, I thought we'd have a quick recap so we're in the same position, or bring up to ourselves thinking about what we mean by allele, and also some of the limitations on that in terms of the makeup of the bird. So first of all then, we know that any new mutation is actually just a gene that has mutated. It never produces a new gene, so there are exactly the same number of genes in a light green as there is in a sky blue as there is in a spangle. It's just that one of the genes of the original wild type bird has mutated to produce that particular variety, and the mutated gene remains in the same locus, so the same place. So the allele of a particular gene, so the allele for blue, is in the same place as the allele for the blue, of the green, so it's in the same place on the chromosome. We also know that the bird inherits one set of chromosomes from each of the parent birds, so it ends up with two sets of chromosomes, which means that it can only ever have two of any particular allele for a gene, whether that gene is mutated once, twice, 3 times, 4 times or 5 times. It can only ever contain 2 allele, it can't contain 3. So what does that mean in terms of an allele that has or a gene that has mutated more than once, and therefore has more than 2 allele for the same gene? We often refer to this as a gene family. So in a gene family there can be a number of allele for the same gene, but as I previously said, just to double check, the bird can only ever inherit 2 of those particular allele, so it can only ever have 2 of those allele. Right, so that's a quick recap on where we are in terms of the bird and the mutations and the number of allele that they can have. The next thing we want to have a look at then is the hierarchical notation for allele, and what I mean by this is what order would we place them in if there was more than one or more than 2 mutations in terms of their dominant to recessive. And we use some symbols to map this out, and the symbols that you might recognise, they're very similar to mathematical symbols, which is greater than or equal to, and those are the symbols similar to what we use, but we use them in a different context. So let's take, for example, the dark factor. So we know, and if we have a look at the bottom here, you can see just there that the dominant mutation has the V pointing to the right, so the one on the left of that V pointing to the right is dominant to the one on the right. So a bit like mathematical mutation is greater than, this one is dominant to. And the codominant is the equivalent mathematical symbol for is equal to. So so we use the two V shapes pointing outwards, so one pointing to the left and one pointing to the right, as we can see down here. Ooh. As we can see down here. So we would write this as follows. So for the dark factor, the wild type is dominant to the mutated, the mutated dominant dark factor is dominant over the non-mutated dark factor, or D is dominant to small d. And we write the similar way with the spangle. So we know that the spangle factor is codominant, so therefore we write it as S is codominant to S. I hope that makes sense. So these symbols, we'll be using a lot more of these symbols over throughout the rest of this video, and as I go through them we will continue to explain them and talk about what that means. So that's the beginning of the way we identify the hierarchical notation for a particular allele. So the family we're going to look at, or the gene family that we're going to look at for this video, is the dilute family, because this family has a number of mutations, and you can see them there. So we've got the wild type, which, as you can tell, I'll put the abbreviation is DIL is what I'm using for dilute, and because that starts with a capital D, we know that the wild type is the dominant allele. So if it contains any of the wild type, you won't produce any of the varieties. So it's exactly the same as any dominant recessive type variety. We've then got the dilute, okay, which is written as DIL, and we've then got a clear wing, which is written DIL at CW, and the grey wing, which is written DIL at GW. Now notice that there are 4 allele within the dilute family, but again, the bird can only have 2 of those allele, or can only inherit 2 of those allele. So it can't be, wild type with a clear wing and a grey wing allele. It can only have 2 possible combinations of those 4 genes. So it's easy enough here for us to understand that the dominant of these is and the recessive relationship to these based as they are. So based on that, so if it only contains those. But we'll need to understand the whole way each of those allele will interact with each other, because the fact that the most dominant is the wild type, we need to understand how the recessive genes to the wild type might interact with each other in terms of their recessive dominant or codominance. And what I always like to do when I'm thinking about this is start off with what I refer to as the pure variety. And by pure variety, I mean it's a concept of for me, it's a concept where the pure variety is where there is no split, or the bird is homozygous for that particular gene. So it contains 2 of the same allele for that gene. So within the dilute family, the wild type would contain 2 of the wild type allele. The clear wing would contain 2 of the clear wing allele, and the grey wing would contain 2 of the grey wing allele, and the dilute would contain 2 dilute allele. And the reason why I always think of those as being the pure varieties, because then it helps me to understand how they might interact and why there might be some strange or apparently strange outcomes when we start to consider putting the various allele onto, or 2 different allele onto the same bird. And we'll have a look at this when we start looking at the Punnett square. So, which is what I want to do now. So let's begin by making our first prediction using the Punnett square, and we'll start with light green split dilute and a light green split grey wing, and see how what the outcome of those birds might be in terms of our varieties. And they'll begin to, when we understand what the outcomes of those are, we'll begin to be able to put some hierarchical order around this particular gene family. So let's pull up the Punnett square, look at the genes that, or the allele that we're interested in, and begin to explore what the outcome might be. So here we are then. Here we can see the Punnett square for this particular bird. As I say, the light green dilute along the top here, or split dilute along the top, and a light green split grey wing along the side. Because they're both birds are light green, and there's no split for blue here, so they just contain the wild type blue gene. We don't need to worry too much about that, because that's the only gene they can pass on. We know whatever we produce is going to be a light green or a green series version of that particular bird. So let's just have a look at the other ones, and you've got the Punnett square here in front of us. If you want to pause the video now and have a go at this yourself, then do so, and I'll meet you back here once you've worked out what you think the various bits is, and we will begin to go through it and have a look at it. So welcome back. I hope you have completed it, and you've got the same outcome as me, and in this case there are 4 different possibilities for the outcome of this bird, and let's just run through them. We know this one here, the top left hand side of the Punnett square, so top left, is going to be a normal green, because both of the allele for the dilute gene are the wild type gene, so this is just going to be a normal green containing no splits whatsoever for the dilute gene. Let's now consider the top right. This one, we've got the wild type gene for dilute, and we've got the dilute gene, and we know that the wild type gene is dominant over all of the other allele, so therefore this is going to be a light green split for dilute. So light green split for dilute. Now let's consider the bottom left, and here once again we've got a wild type gene plus the dilute grey wing gene. I'll just amend that to say with a low D. And again we know that the wild type gene is dominant over the mutated dilute grey wing, and therefore that's going to be a light green split grey wing. And let's now consider this next one here. Well, here we've got in effect 2 recessive genes, and this is the first time in this series we've come across where we've got 2 recessive genes in the same bird on the same for the same, or 2 recessive allele in the same gene on the same bird. So we've got a dilute and a dilute grey wing. So we need to understand what the potential outcome of that bird is in terms of the hierarchical order of the gene family. So let's snip back now. We'll come out of this and we'll have a look at what this means. Okay, so we're beginning to explore the hierarchy for the dilute gene family, as I say, and here we can see it. So we know that wild type is dominant over the dilute allele. We know that the wild type is dominant over the grey wing allele, and in terms of the grey wing and dilute interaction, then the grey wing is dominant over the dilute. So far you can see where I've written it at the lower part here, we can see that it goes, in terms of the hierarchical order, is dilute is dominant over grey wing, and grey wing is dominant over dilute. So let's go back to the Punnett square now, and let's try a different two birds. We're going to try one of the birds that was the outcome from the last pairing, where we're going to have a grey wing split dilute, or a light green grey wing split dilute, and we're going to have a light green split clear wing. So we've got a grey wing split dilute and a normal split clear wing. See if you can identify the various allele that each bird has, and see if you can fill out a Punnett square. And then once you've done that, so if you pause the video and do that, once you've done it, you can unpause the video, and I'll have my completed Punnett square up on the screen. So here we go, I've completed the Punnett square for our light green grey wing split dilute and our light green split clear wing bird. And as you can see, we've got a number of outcomes here, and once again we've got 4 different outcomes. Some of those we'll begin to be able to understand, and one of those is going to be slightly different. So in terms of the top two, so the top left and the top right, in both of those cases we've got the wild type, or we've got at least one of the wild type allele, which means that the bird is going to be a wild type bird, so a light green. And in the top left it's going to be light green split grey wing, and the top right is going to be a light green split dilute. So those are easy enough to understand. If we come down here to the dilute, we've got a bird on the bottom right hand corner which is a clear wing, and it contains a clear wing allele and the dilute allele. Now, a bit like the grey wing and the dilute, the clear wing is dominant over the dilute. So in this case you're going to have some clear wing birds that are going to be split for dilute. The final one is the really interesting combination, which is a dilute grey wing and a dilute clear wing. So one allele for the grey wing and one allele for the clear wing, and that actually has a codominant reaction. So it's a codominant hierarchy. So we neither produce a grey wing or a clear wing. What we'll do now is we'll pop back over, we'll close this and reopen the presentation, and we'll have a look at that particular bird and what that means in terms of the hierarchical order. So here you are then, we're back at the presentation, and I've reconfirmed what we said just now. So the wild type is dominant to the dilute, the wild type is dominant to the grey wing, the grey wing is dominant to dilute, and the clear wing is dominant to dilute. So we can see from this that the wild type is dominant to everything, and dilute is recessive to anything. So dilute is completely recessive. So from that we would know if it's completely recessive to all of the other allele, in order to get a dilute bird you must have 2 of the recessive dilute allele on the same bird. So 2 recessive dilute allele is the only way that you will produce a dilute. So let's think about this, the bird that we come out with, which was a dilute grey wing and a dilute clear wing, or a grey wing and a clear wing bird, and we said they were codominant. So let's have a look at what that now does to our hierarchy. So you can see the bottom one there, the dilute, the clear wing and the grey wing are codominant, and they actually produce a bird that's known as a full body colour grey wing. Normally the grey wing has a diluted body colour, and the clear wing which has clear wings but a full body colour. In this case we're going to get a bird with full body colour but with the markings of the grey wing. So it contains the body colour of the clear wing but the wing colour or the wing formation and pattern of the grey wing. So we can see that it's an in-between bird, in between those two, as most or all codominant varieties are. And right at the bottom we can see now that we have the full hierarchical order for the dilute family, and we can see it goes wild type is dominant to grey wing, clear wing and dilute. Grey wing and clear wing are dominant to dilute, and that clear wing and grey wing are codominant. We could put those two round in either order. We could have put the clear wing in front of the grey wing, or the grey wing, as we've done in this case, in front of the clear wing. That doesn't matter because we know they are codominant. So I hope that makes sense in terms of the hierarchical order for this particular gene family. In part 8 we'll revisit this in terms of the blue family, because understanding this hierarchical order helps us to begin to understand how the yellow face and the blues interact with each other, and what the hierarchical order for that is, and how we might then predict or expect to predict an outcome. So I hope that's okay. Please do play around with these particular allele. Have a look, get your Punnett squares up and play around with it and see if you can predict the outcome of various birds. In part 7 we're going to look at gene linkage. We already looked at gene linkage when it was on the sex chromosome, but gene linkage on the other chromosomes and how that works. And during that process we will of course revisit the idea around the opaline cinnamon conundrum and how we can get those two onto the same chromosome. So that's what we'll be covering in part 6. Final word on the gene hierarchy within a particular gene family. Don't forget, you cannot get more than 2 allele on a particular bird, so you can't get a full body colour split dilute. So if I go back to here, you can't have a bird that is a full body colour grey wing split dilute, because for a full body colour grey wing it needs to have a clear wing allele and the grey wing allele, and if it was then split dilute it would contain 3 allele for a particular gene, and that is not possible. Right, that's it then for this particular video. Hope you enjoyed that. If you did, don't forget to hit the like button. Please do subscribe, and I will see you when we cover gene linkage.